Hello students, today we embark on an exciting journey into the unseen microbial world as we explore the general characteristics and biological role of the microscopic world filled with fascinating entities like phages, viroids, mycoplasma and E. coli, organisms that shape ecosystem, drive evolution and influence both health and disease. By exploring their unique structures, life cycle and biological role, we uncover how these tiny agents impact agriculture, medicine and biotechnology on a global scale. During today's discussion, we will cover these learning objectives. To understand the general characteristics, structural features and life cycle of bacteriophages, viroids, mycoplasma and E. coli, emphasizing their diversity and unique biological properties. Explain the biological role of phages, viroids, mycoplasma and E. coli in natural ecosystems, agriculture biotechnology and human health, including their beneficial and pathogenic aspects. Analyze the mechanism by which these microorganisms interact with their hosts, including infection cycles, gene transfer and contribution to the genetic diversity and disease. Evaluate the economic, medical and ecological impact of these microorganisms, such as their role in crop disease, antibiotic resistance, environmental indicators and therapeutic applications. Bacteriophages. The characteristic features of bacteriophages are: the bacteriophage are also known as phage, most abundant organism found in all environments where bacteria exists, such as soil, ocean, intestines, etc. Bacteriophages were independently discovered by Frederick William Todt in 1915 and by French-Canadian microbiologist Félix de Héroulé in 1917, coined the name bacteriophage, bacteria eater or virus that kills bacteria. Bacteriophage structure. Bacterial viruses are called as bacteriophages or simply phage and have been intensively studied as model systems for molecular biology and genetics of virus replication. Bacteriophages occur in more than 140 bacterial genera and in many different habitats. Phages are much smaller than bacteria, typically between 24 to 200 nanometer in length. A bacteriophage is made up of a protein coat known as capsid, which encapsulates the genome. It consists of a polyhedral head. It may be enveloped or non-enveloped and have different shapes such as rod-shaped, filamentous, isometric, etc. The capsid is made up of many capsomeres. The size and the shape vary in different species. Many phages contain double-stranded DNA, others contain double-stranded RNA, single-stranded RNA or single-stranded DNA. They don't possess cellular structures and metabolic machinery. They need a host for reproduction. The genome codes for a protein ranging from 4 to 100. MS2 bacteriophage genome codes for 4 protein. The largest genome found in bacteriophage is 735 kilobase pairs. Many phages contain specialized syringe-like structures or tails that bind to receptors on the cell surface, bacterium, and inject the phage nucleic acid into a host cell. The tails may be long or short, contractile or non-contractile. Tail fibers are present. It helps in anchoring the virus to the bacterial cell wall. The classification of bacteriophages. Phages can be broadly classified on the basis of their life cycle as lytic or the virulent phages. They immediately hijack the host's machinery to produce progeny phages, end with lysis or the destruction of the bacterial cell wall, releasing multiple new phage particles. Example, T-even phages such as T2, T4 or T6. Second is the lysogenic or temperate phages, which integrate their genome into the host's chromosome as a prophage, replicating passively along with the host. Under stress conditions, the prophage may re-enter the lytic cycle, destroying the host cell. Example, the lambda phage in E. coli. Replication cycles in bacteriophages. First is the lytic cycle. The step 1 in lytic cycle is the attachment. The tail fibers attach to specific receptors on the bacterial cell wall. Then penetration occurs. The tail injects phages nucleic acid into the host cytoplasm. Biosynthesis. The host's machinery is redirected to synthesize phage components, DNA proteins and enzymes. Assembly. New phage particles are assembled within the host cell. Lysis. The bacterial cell bursts, releasing dozens or hundreds of progeny phages. The lysogenic cycle includes the integration. The phage DNA integrates into the bacterial genome, forming a prophage. Then comes the replication. The prophage is replicated along with the host DNA during cell division. Then comes the induction. Environmental signals, for example, the UV light, stress may trigger the prophage to excise and enter the lytic cycle. Biological roles of bacteriophages. They serve as the agents of natural biocontrol or bacterial population control. The number of bacteria in ecosystem, for example, the soil, gut and seas. As phage display technology and gene delivery vectors, they are employed as potential genetic engineering tools. They are used as therapeutic agents. Phage therapy thereby acts as a substitute for antibiotics, particularly in fight against microorganisms that are resistant to many drugs. Antimicrobial resistance, one of the biggest risks of global public health and development, may be controlled by phage therapy. They are used in recombinant DNA technology like transduction, a process that transfers gene across bacteria and adds to genetic diversity, and for horizontal gene transfer. They are used in the food industry to kill bacteria in meat or cheese products. Next comes the viroids. Characteristic features of viroids. It was first examined in the potato spindle tuber viroid that caused a huge loss to the potato industry. Viroids were first discovered, identified and named by Theodor Otto Diener in 1971. Viroids don't possess protein coat or the capsid or envelope. They are naked RNA molecules. Viroids are infectious agents that affect only plants, therefore are also called as plant pathogens. They are the smallest known infectious pathogens, even smaller than viruses. Viroids consists of short, circular, single-stranded RNA molecule, typically 250 to 400 nucleotides. Viroids are composed solely of RNA, which is much smaller molecule than the genetic material found in viruses. Viroids are the nucleic acid entities of relatively low molecular weight and unique structure. These entities hijack the cellular machinery present in the plant cell to replicate new copies of themselves. Viroid RNA does not code for any protein. It exerts its effect through interaction with host machinery. Viroids can be transmitted through contact and infected plants, contaminated equipments or insects. The structural uniqueness and the evolutionary significance of viroids are considered living fossil that may represent remnants of an ancient RNA world. This small, non-coding RNA genome resembles small self-replicating molecules hypothesized to have existed before the evolution of DNA and proteins. This makes them important for understanding the origin of genetic material and early life evolution. Their rod-like RNA structure confers exceptional stability, enabling viroids to persist in plant tissues and resist degradation by cellular enzymes. Additionally, viroids interfere with the host's normal gene expression through RNA silencing mechanisms, leading to disease symptoms. The classification of viroids. Viroids are grouped into two major families. The Pospeviridae replicate in the nucleus of host cell. Example, potato spindle tubal viroid, PTSVD, and the citrus exocotis viroid, CEVD. Avsin viroidae replicate in chloroplasts. Example, avocado sunblot viroid. Replication and pathogenesis. Viroid replication occurs through a rolling cycle mechanism. The viroid RNA uses the host's DNA-dependent RNA polymerase II for replication. New viroid RNAs are synthesized, processed, and circularized. The replication process does not produce any proteins. Instead, viroid RNA itself is the infectious entity. Pathogenesis. Interference with the host gene expression. Viroids can act like microRNAs, binding to complementary plant RNAs and disrupting the normal gene regulation. Alterations in the plant physiology, resulting in stunting, leaf curling, chlorosis, and decreased fruit quality. Transmission and spread. Viroids are highly transmissible within the plant populations. Mechanical transmission via contaminated tools, pruning and grafting, seeds and pollens transmission. Some viroids can spread through reproductive structures. Insect vectors. Certain insects may inadvertently transfer viroid RNA between plants. Dear students, let's now talk about the biological roles of viroids. Viroids are responsible for serious plant diseases affecting agricultural productivity and economy. CRISPR-Cas-based viroid diagnostics are being explored for ultra-sensitive field detection. Nanopore sequencing enables the full-length viroid genome analysis in real time. Viroids are major threats to global agriculture, especially potato, tomato, citrus, avocado, and grapevine industries. The estimated crop losses in some countries exceed billions of dollars annually due to viroid outbreaks. Management and control strategies. Viroid resistance to heat and chemical treatments make eradication difficult, necessitating stringent agricultural hygiene practices. Hygiene practices include the regular disinfection of tools and machinery. Resistant cultivars. Breeding of viroid resistance is an active area of research. Rapid diagnostics. Deployment of sensitive molecular detection tools to identify the infections early. We will now discuss about mycoplasma. The characteristic features of mycoplasma. Mycoplasma comes from two words. Myco means fungus-like, forming branching filaments, and plasma, denoting their plasticity of shape, are free living in environment. Knockert and Rocks were the first in 1898 to isolate mycoplasma as a filterable and highly pleomorphic microorganism from bovine pleuropneumonia. Electron microscopic image of Mycoplasma bovis colonies appeared as fried eggs, central dark zone surrounded by a lighter peripheral zone. Mycoplasma are known as pleuropneumonia-like organism, PPLO, or mycoplasma-like organism, MLO. Facultative anaerobic pleomorphic, also found in soil sewage, osmotically unstable. They are self-replicating on Artificial culture media. Resemblance to certain virus properties. Mycoplasma are very small, 150 to 350 nanometer in size. They are filterable in bacterial filters. They lack cell wall around their cell membranes that makes them resistant to antibiotics targeting cell wall. Differences from virus properties. Mycoplasma are free living in the environment. They can grow on the artificial culture media. They lack a rigid cell wall, which is replaced by a triple layered cell membrane containing sterol. They are completely resistant to antibiotics acting on cell wall such as beta-lactams, penicillin. They are highly pleomorphic, exist in coccoid, filamentous or even in helical forms. They are poorly gram-negative, better stained by Giemsa stain. Mycoplasma reproduce by binary fission and budding, non-sporing and non-flagellated, usually non-motile. They lack nucleus and other membrane-bound organelles. Genetic material is single DNA duplex. Mycoplasmas are common contaminants of continuous cell cultures, thus interfere with the growth of viruses in cell cultures. Structural and functional uniqueness. Cell wall absence makes them inherently resistant to antibiotics targeting the cell wall synthesis such as penicillin and other beta-lactam drugs. Membrane composition. Incorporation of sterols from the environment strengthens their otherwise fragile membrane. Pleomorphism. Shapes range from coccoid and filamentous to helical forms, enabling them to adapt to varying environments. Contaminants in the cell culture. Mycoplasma are notorious contaminants of laboratory cell culture, interfering with the experimental results and virus propagation. Pathogenesis and diseases caused by mycoplasmas. Mycoplasmas cause a range of diseases in humans, animals and plants, respiratory infections. Mycoplasma pneumoniae causes atypical pneumonia, often called as the walking pneumonia, characterized by mild symptoms and prolonged cough. Urogenital infections, Mycoplasma hominis and ureaplasma urealyticum are implicated in pelvic inflammatory diseases, infertility and preterm birth. Opportunistic infections often infect the immunocompromised individual. The cell culture contamination. Mycoplasmas disrupt the laboratory experiments by altering the host cell metabolism and gene expression. We will now talk about the resistance and adaptation of mycoplasma. Antibiotic resistance, intrinsic resistance to beta-lactam antibiotics due to the absence of cell wall. Environmental flexibility, they survive in osmotically unstable environments by adjusting membrane sterol content. Immune evasion, they exhibit antigenic variation, enabling persistent infections in hosts. Control and prevention, hygiene measures, regular sterilization of laboratory equipments and strict aseptic techniques. Antibiotic therapy, tetracyclines and macrolides are effective against mycoplasma infections. Monitoring in cell culture, routine screening to prevent the contamination. Vaccinations, vaccines available for some veterinary mycoplasma diseases. Dear students, we will now move on to the biological roles of mycoplasma. Certain mycoplasmas are part of the gut microbiota and may contribute to the overall health of the host by influencing the composition and function of the gut microbiome. Some mycoplasmas engage in beneficial symbiotic relationships with their host, potentially aiding in nutrient acquisition and provide protection against their pathogens. Mycoplasmas play a role in soil ecology by participating in nutrient cycling and decomposition processes. They contribute to the genetic exchange and variability in the microbial communities. We will now talk about E. coli. Characteristic features of Escherichia coli or the E. coli. E. coli is the part of normal microflora of the human body and the most studied bacteria due to its multiple potential characteristics. E. coli was discovered by Theodor Escherichia in 1885 after isolating it from the feces of newborn. Most of them are harmless and opportunistic, but few are causing different intestinal infections like traveler's diarrhea. The primary habitat of E. coli is in the gastrointestinal tract of humans and many other warm-blooded animals. It colonizes a newborn's gastrointestinal tract within hours after birth and even helps to keep our digestive tract healthy. E. coli is classified into serotypes or 150 to 200 serogroups based on 3 antigens: somatic or cell wall antigen, capsular or K antigen, and flagellar or H antigen. The intestinal pathogenic E. coli strains are divided and classified into several pathotypes. The first one is the enterotoxigenic, the second one is the enteropathogenic, the third one is the enteroaggregative, and the fourth one is the Shiga toxin producing E. coli, and the fifth one being the enteroinvasive E. coli. As Shiga toxin producing E. coli can cause severe food-borne diseases, it is transmitted to humans primarily through the consumption of contaminated foods such as raw or undercooked ground meat products, raw milk, and contaminated raw vegetables and sprouts. Raw vegetables or vegetable salads, uncooked meat, unpasteurized milk, etc. are dangerous sources of E. coli that can lead to different illnesses. Humans are most likely to be infected with E. coli O157 H7 found in cattle. E. coli is gram-negative bacterium, appears as short straight rods under microscope or the bacillus-like. They are facultative anaerobes and can grow in both the presence as well as the absence of oxygen. Many strains possess peritrichous flagella, allowing the motility. They have the doubling time of 20 minutes under optimal conditions and can be cultured very fast as compared to the rest of the microbes. Its genome size is approximately 4.6 million base pairs with around 4,000 to 5,000 genes. They are mesophilic microorganisms with optimal growth temperature is around 37 degrees Celsius, suitable for human body. They are cultured on nutrient media like nutrient agar. We will now see the biochemical characteristics of E. coli, which include the oxidase, which is negative, catalase, which is positive, and indole, which is also positive. E. coli acts as indicator of fecal contamination in water and food. It is widely used in biotechnology and genetic engineering due to its well understood genetics. Let us now see about the ecological niche and distribution of E. coli. In the normal habitat, it is commonly found in the lower intestinal tract or the colon of humans and warm-blooded animals. It colonizes infants within hours of birth, playing an important role in the development of gut microbiota. Environmental survival of E. coli. Though primarily an intestinal commensal, E. coli can survive temporarily in soil and water, serving as a key organism in environmental microbiology. Biological roles of E. coli. E. coli is the most widely studied prokaryotic model organism and an important species in the field of biotechnology and microbiology, where it serves as a host organism for recombinant DNA and experimental workhorse for DNA manipulation and protein production. E. coli is a part of the gut microbiota and helps the digestion and synthesis of vitamin K. It is one of the important members of normal microflora of human body. It is employed as a model organism in study, particularly in the field of genetics, molecular biology and biotechnology, as well as in water analysis. When fecal contamination is detected in food or water sample, it can be used as a marker for prospective contamination and health hazards. Diarrhea, UTIs and the newborn meningitis are among the illnesses caused by some strains such as EHEC and UPEC, a recombinant DNA technology tool that inserts foreign genes into E. coli to produce insulin hormones and enzymes. Several strains of E. coli have been identified as good and effective probiotics and are currently employed in pharmaceuticals. Dear students, in summary, phages, viroids, mycoplasma and E. coli represent the remarkable examples of microbial diversity, each influencing the health and ecosystem in unique ways. By understanding their structures, life cycles and biological roles, we gain the insights into their significance in medicine, agriculture, biotechnology and environmental sustainability. These microscopic agents not only pose challenges as pathogens, but also offer opportunities as a tool for innovation, therapy and scientific discovery. As we conclude, let us appreciate how studying these organisms deepen our knowledge of biology and equips us to address the real-world problems through science and technology. Thank you.